Hello guys, welcome to my YouTube channel. The topic for discussion is the nerve muscle physiology. This is the basis for understanding the central nervous system. In this video, we will cover the cells of central nervous system, types of neurons, myelin, and the nerve conduction velocity. Now, starting from the basic, we call something as functional unit of central nervous system. What are these functional units? Obviously, these are the neurons or nerve fibers. Remember guys, both of these can be used interchangeably, but for conceptual understanding, neurons are the structures present in the central nervous system, while as the nerve fibers are the structures present in the peripheral nervous system. First, we are going to discuss the importance of the nerve fibers, but before that, let's understand the cells found in the central nervous system. We have the most important ones are the neurons, and we have something supporting cells for the neurons called as glial cells. So out of these two, which are actually more, neurons or the glial cells? Actually, the glial cells are more, having a ratio of 1:10. That for every neuron, there are 10 glial cells or the 10 supporting cells. These glial cells are very good friends of the neurons and are very important supporting system of the body. Now let's have a small discussion on these glial cells. Broadly speaking, they can be classified into the two types. Firstly, the macroglial cells and microglial cells. We should remember that these microglial cells are different from the rest of the glial cells in terms of different embryological origin. These are derived from the monocyte, macrophage lineage, and these are the phagocytic cells. Now coming towards this side are the macroglial cells. They can be star-shaped astrocytes, or the cells having very few dendrites called as oligodendrocytes, or they can be schwann cells. Out of these macroglial cells, out of the three, the astrocytes are very important from the rest of the cells. Now let's take a small discussion on these astrocytes. These astrocytes can be further subdivided into the two types. Firstly, they can be protoplasmic astrocytes and they can be fibrous or fibrous astrocytes. These protoplasmic astrocytes are only seen in the gray matter, while these fibrous astrocytes are seen in white matter. These astrocytes show a very important intermediate filament known as GFAP or glial fibrillary acidic protein. This intermediate filament is considered to be a tumor marker for the tumors of astrocyte origin. tumor marker for tumors of astrocyte origin or astrocytomas. So this intermediate filament is the tumor marker for the astrocyte origin or astrocytomas. Now let's first understand some of the function of these astrocytes. The most important function of the astrocyte is BBB, or the blood brain barrier. The astrocytes help in the formation of the blood-brain barrier. But what is the meaning of this blood-brain barrier? Consider this a brain tissue. Obviously, it will be perfused by some of the blood vessels. Brain tissue and blood vessels. The substance cannot cross easily between the blood vessel and the brain due to the presence of BBB, or the blood-brain barrier. This blood-brain barrier is formed most importantly by the capillary endothelial cells. There are the capillary endothelial cells. These endothelial cells form the tight junctions there. These tight junctions are actually reinforced by the food process of these astrocytes. For example, this is the astrocyte. This is its food process. It goes here. And this way, astrocytes help in the formation of blood-brain barrier. The second function of these astrocytes is the synaptic clearing. Synaptic clearing. Following signal, there is effective uptake of substances like potassium is there, neurotransmitters, by the astrocytes. The effective uptake of these substances is very important for effective functioning of neurons. Following any signaling event, some of the K positive is liberated, which is taken back by the astrocytes. Certain neurotransmitters like, for example, glutamate is there. It's taken by the astrocytes. This way the synapse is cleared and is very important for the effective functioning of the neurons. Now, as the astrocytes have discussed it, now we are going to discuss the other macroglial cells like the oligodendrocytes and schwann cells. Oligodendrocytes and the schwann cells offer friendship to neurons just by hugging them in the form of myelination. So they play a role in myelination. In central nervous system, myelination is done by the oligodendrocytes, while in peripheral nervous system, myelination is done by schwann cells. So this is the difference between them. In central nervous system, myelination is done by oligodendrocytes, while in peripheral nervous system, myelination is done by schwann cells. The most important difference between these two is that the oligodendrocytes can perform multiple myelination. That is, the single oligodendrocyte can myelinate multiple neurons. On the other hand, S for S, that's for schwann cell, single myelination. that is the ratio is 1:1, that one Schwann cell can myelinate only one nerve fiber. Oligodendrocyte, multiple myelination in CNS, Schwann cells, single myelination in peripheral nervous system. Now we have completed the glial cells, and now we will move on the neuron structure and the types of neurons. Now first we will see the types of neurons. Firstly, there is the unipolar neurons. Actually, we have here a cell body and a one process. Cell body and one process. For example, if we look at the example, it's found in lower invertebrates. Then we have pseudo-unipolar neuron. Here we have a cell body and a one process, but the process divides into two. One goes into the spinal cord and other goes to the skin. It's found in, for example, dorsal root ganglion neurons. Then we have bipolar neurons. Here we have bi means two. We have two processes. This is a cell body, this is a one process, this is another process. Here this is a dendrite, and here this is the axon. It receives and it conducts. Where it is found, for example, it's found in retina. It's found also in olfactory epithelium. And at last we have multipolar neurons. Among all these types, multipolar neurons are most numerous in number. Here we have multiple processes. If this is the cell body, there are multiple processes, but we should remember there is only one axon. There are many processes, but we should remember there is only one axon. It's found in, for example, in spinal, alpha motor neuron. So these were some types of the neurons, and now we will see the structure of the neuron. Now the structure of neuron. Firstly, I will draw the cell body here. This is the centrally placed nucleus. This is the axon, and here this is the synaptic terminal or the end point of the axon. What are these? These are the myelin covering. And here are the nasal bodies. This is the nucleus. We have some dendrites here. Here there are multiple dendrites are. And here they are nasal bodies. What are these nasal bodies? Actually, these are the rough endoplasmic reticulum and help in the formation of proteins. Protein formation. Here, the most important concept is this and this. This is axon hillock. We should remember this axon hillock, and this is the initial segment. We should remember that the axon hillock is a part of cell body and initial segment is a part of what? Axon. This is the axon. It helps us in conduction. this is myelin. What does myelin does? We will discuss it later. Firstly, if the myelin is present, there we have fast conduction. If myelin is absent, we have slow conduction. We will discuss it later. And here, where the myelin is absent, it is called as node of Ranvier. We should remember this N. N stands for no myelin. And secondly, sodium channels are maximum here. How much? About 2000 to 12,000 per square meter. And this is the synaptic terminal. So we should remember this. Axon hillock is a part of cell body and initial segment is a part of axon. And this is the cell body, also called as soma. So this was the structure of neuron. To conduct an impulse, we have to first generate the action potential. But where does it generate? In motor neuron, it is generated in or initiated in the where? Axon hillock plus initial segment. And in sensory neuron, it is initiated in where? First node of Ranvier. So in motor neuron, it's initiated in the axon hillock plus initial segment, while in sensory neuron, it's initiated in the first node of Ranvier. Though this axon hillock has the name axon in it, but it is a part of soma or cell body. That's why we say naam mein kya rakha hai. Now comes the very important topic, axon conduction. The information in the axon can be conducted from cell body to synapse, or it can go from synapse to cell body. The first one is called as antegrade axonal transport, and the second one is called as retrograde or reverse axonal transport. Intrograde axonal transport is by the help of protein known as kinesin, and retrograde or reverse axonal transport is by the help of dynein protein. This antegrade axonal transport can be fast or slow. Can be fast, about 400 per day, or slow. But this retrograde reverse transport is always fast, about 200 millimeter per day. We should remember during viral invasion, the viruses prefer retrograde or reverse axonal transport. For example, during rabies, the rabies virus, or herpes virus. Even the tetanus toxin prefers retrograde or reverse axonal transport. Now we will discuss something about myelin. It is the combination of myelin is the combination of lipids and proteins. Proteins are about 20%, while the lipids are about 80%. What type of lipids are there? They are named as sphingomyelin, and the proteins are named as myelin basic proteins or MBP. These myelin basic proteins have the basic nature, that's why they are called as basic proteins. They are also named as zero proteins. What is important here is that sometimes this myelin basic protein or MBP is destroyed by autoantibodies, which leads to the demyelination and which causes decrease in nerve conduction, and it leads to a particular disease known as multiple sclerosis. So what happened in multiple sclerosis? There occurs the decrease in nerve conduction due to the demyelination, which is caused by the autoantibodies destruction the myelin basic protein. So this was all about the myelin, and now we will see about the nerve conduction velocity or NCV. Now we will see about the nerve conduction velocity. Firstly, we will see the factors on which it depends. Factors. Firstly, it depends on the diameter of the neuron. Diameter of neuron is directly proportional to the nerve conduction velocity, which means increase in diameter is directly proportional to increase in nerve conduction velocity. Secondly, it depends on myelin. Which increases the nerve conduction velocity. How it increases the nerve conduction velocity? Firstly, increasing the thickness. Secondly, what it does, it increases axonal resistance, which leads to what happened there. There is no diffusion out of ions, and the impulse go to nearby node of Ranvier. Third, temperature. Temperature is also directly proportional to the nerve conduction velocity, which means increase in temperature is equal to increase in nerve conduction velocity, and decrease in temperature leads to decrease in nerve conduction velocity. Before going to the fourth factor, we have to see the membrane capacitance. It means the ability to store the charges. The membrane capacitance, this membrane capacitance is inversely proportional to thickness. For action potential or the nerve conduction, we don't need to store the charges, we need the free flow of charges. Therefore, the low the membrane capacitance, the high the nerve conduction velocity. The fourth point is this myelin increases thickness. We know it increases thickness. If this is axon, this is the myelin, it increases its diameter, it increases its thickness. So myelin increases thickness, which leads to decrease in membrane capacitance, which further leads to increase in nerve conduction velocity. So guys, this was all for today. Hope you enjoyed this video, and if you do, then drop the thumbs up and share it with your friends. See you in next video. Bye.